Top 10 Viral Facts About Viruses. In a world brimming with remarkable things, infections may very well be the most abnormal. They are personally engaged with the existences, all things considered, yet they may not in fact be alive themselves. However, as the most recent year has demonstrated, they can effectively affect living things. A solitary infection can develop dramatically into a world-changing event. Here are 10 major realities about these small, irresistible things. Number 10. Are viruses alive? You may feel that the discussion on what establishes life is settled. By and large we realize if something is alive just by taking a gander at it, yet infections obscure the lines. They are made of the things we partner with life, proteins, lipids, and nucleic acids. They even do things we partner with life, they imitate and develop. Yet, the key actuality is that they can't do it all alone. For an infection to imitate it should taint a host cell and capture their digestion. Most researchers consider infections as non-living gatherings of synthetics that are truly adept at making duplicates of themselves. There are a few specialists, however, that consider infections as particularly alive. They refer to the intricacy of their genomes and the speed of their development. They say that when we picture infections in their total structure, as little containers of DNA or RNA, we can envision they are dead. In any case, just similarly a bacterial spore is dead when it is latent. When an infection enters a cell and starts the intricate assignment of making more duplicates of itself, they think it is completely right to consider them living organisms. Whichever side of the discussion you take, the life pattern of an infection is an interesting one. Number 9. Viruses might be the inception of life. Advancement is the most ideal method of seeing how life on Earth has changed and grown. However, numerous individuals guarantee that development can't reveal to us anything about the root of life itself. One speculation anyway utilizes development to clarify how life, as far as we might be concerned, may have appeared. In the first place there was RNA, a particle firmly identified with DNA. Critically, however, RNA can wind and shape subatomic hardware that can make duplicates of itself. When the main RNA particle that could do this framed, it would quickly multiply. Assuming changes built up that improved it at making more duplicates, it would outcompete other RNA strands. In this manner lifeless particles can evolve. What are the least difficult things that utilize RNA and DNA to imitate? Infections. In the virus world speculation, it was infections that started things out, and those that could contaminate living beings are the ones that have been made do until now. Number 8. You are generally infection. Infections are all over. Any place there is life, there are infections that are related to them. While some are truly recognizable through their impacts on us, many are innocuous to the point that we never realize they are on us. Furthermore, there are a ton of us at any one time. We will in general consider people a mass of our tissues and cells, the pieces that share our DNA. In the human body there are around 10 human. This number is little anyway contrasted with the microorganisms that live inside us and on us. There are tenfold the number of bacterial cells in any human as there are human cells. At that point there are the infections. Every human conveys multiple times more infections than they do human cells. While a portion of these infections will target human cells, large numbers of them need just to taint the microorganisms we share our bodies with. Number 7. Viral antibiotics. How since quite a while ago failed to remember infection could assist us with settling the antimicrobials emergency. Probably the best danger in present-day medication is antimicrobial obstruction in microscopic organisms. At the point when our anti-infection agents neglect to eliminate bacterial diseases, we are practically back in the pre-present day age when a straightforward scratch could prompt your passing. Infections might be our saviors. Bacteria are prey to infections very much as are we. Phages are infections that taint microbes. When a phage has tainted a bacterium, it duplicates hundreds or thousands of duplicates of itself, and parts of the bacterium open, heaving more infections to contaminate more microorganisms. On the off chance that you can discover a phage that chases and murders a risky bacterium, that you may simply have discovered an answer for antitoxin resistance. Lots of exploration is being done on phage treatment, as these medicines are called, and numerous researchers are energized by the chance of novel remedies for bacterial contaminations. However, utilizing phages isn't totally new. At the point when a cholera scourge struck India in 1926, specialists took stool from individuals who had startlingly recuperated from cholera and offered it to the wiped out. A significant number of those treated by this fairly grim technique recuperated. Likely the appropriate response lay in phage infections that focused the cholera germs. Number 6. Nobel Prizes. Assuming you need a Nobel Prize, probably the best spot for you to begin your work may be on an infection. In 2020, analysts were granted Nobel Prizes in medicine for their disclosure of the hepatitis C infection. The granting of Nobel Prizes for virology returns a lot further, however. Viruses were first found in 1892 when Dmitry Ivanovsky found that tobacco plants could be contaminated by sickness when infused with a liquid that had been gone through a channel excessively little for any microscopic organisms to go through. The concealed irresistible specialist was before long named an infection, from the Latin for poison, or a disgusting fluid. Some idea that an infection was something fluid. However, Wendell Stanley figured out how to detach the tobacco mosaic infection and clean it into gems for study, demonstrating infections were particles. He won the Nobel Prize for his work in 1946. Since then, popular examination has won the Nobel Prize many occasions for either relieving a viral contamination, like the production of the yellow fever antibody, or working out how infections like papillomavirus causes cervical malignant growth. Number 5. Mind-boggling numbers. It is difficult to check the quantity of infection particles on Earth at any one time. When you checked them, they would have duplicated and been obliterated many occasions over. Researchers can anyway make a decent gauge. These reach from 10 to the force 30 to 10 to the force 32, so many would say there are around 10 to the force 31 on Earth. That is 1 with 31 zeros after it. To place it in context, there are a simple 10 to the force 21 stars in the discernible universe. All of these are in the world, yet we can't see them. This makes it clear exactly how little infections really are. So what might occur in the event that you took some extremely fine tweezers and lay every one of these infections start to finish? A decent normal size of an infection is around 125 nanometers, billionths of a meter. So partition the quantity of infections by their normal size to discover how far our chain of infections would stretch. The answer is that our viral chain would be 800 million light-years long. This would pass past the closest system, and past the adjoining groups of worlds. Number 4. Tiny viruses. We've seen that infections can be little, however exactly how little would they be able to go? That relies upon their nucleic acids. An infection is, fundamentally put, simply an extravagant shell around some DNA or RNA. The shell helps the DNA or RNA get into a host cell, and the nucleic acids power the host cell to make more shells so the infection can spread. The number of qualities can that take? For the circoviruses that taint pigs, all they require are 3 qualities. The whole length of their genome is only 1726 base combined since a long time ago, contrasted with a human's genome of more than 3 billion base sets. With only 3 qualities, the infection can attack a cell and recreate. With a short genome, it just necessitates a little code to house it. The circoviruses are just 17 billionths of a meter across. Some scientists are attempting to get much more modest. One gathering has declared they made a fake infection from pieces of protein and DNA that is 12 billions of a meter long. It is trusted that more fake infections may have a job in cutting-edge clinical medicines. Number 3. Giant, reanimated, viruses. Not all infections are substances to remain little in any case. Some of them can get quite enormous, on the off chance that you think about microorganisms huge. At the point when specialists went searching for microbes in a cooling tower, they gathered what they thought were microorganisms, however they couldn't distinguish them. It was just a lot later that they were found to be an altogether new kind of Goliath infection. They considered it a mimivirus as it was a microbe mimicking infection. Its genome is more than 12 million bases long, and the infection is bigger than the littlest bacteria. Since then, other monster infections have been found, some of them in far-fetched places. In 2014, the biggest ever infection was found in the frozen tundra of Russia. The examples it was found in were more than 30,000 years of age. At that point they did the conspicuous thing and attempted to revive them. The researchers presented amoebae to the thawed-out infections as they probably are aware other monster infections contaminate these single-celled life forms. Notwithstanding being from the Stone Age, the infections effectively attacked the cells and recreated. The capacity of infections to stay dynamic after so long has prompted some hypothesis that liquefying permafrost may deliver since a long time ago failed to remember human microorganisms. Number 2. Viruses on viruses. Mama virus is another Goliath infection that was found in a cooling tower, tainting amoebae. However, what most energized researchers was the disclosure of another infection related to it. This a lot more modest infection targets memivirus and goes after it like a parasite. Due to this, analysts named it Sputnik, and the other infections that assault infections are known as satellite viruses. Sputnik can't taint a one-celled critter all alone and imitate. It can just repeat an amoebae that have effectively been tainted by a mama virus. Rather than straightforwardly commandeering the phone zone apparatus, Sputnik utilizes the infection's replication proteins to make duplicates of itself. Not even infections are invulnerable to the treacherous and bright strategies for viral attack. Number 1. UR-part infection. People all convey infections of some kind or other inside us, however we additionally have infections caught inside our genome. Some infections are not substance to just recreate in our cells. Some utilize subatomic machines to cut our DNA and supplement their own into it. Assuming they do this in a sperm or egg, their DNA can be passed down the ages. More than a long period of time, this has occurred so frequently that around 8% of any individual's whole genome is made of these infections that have gotten fossilized inside us. The infections in genomes can even be utilized to follow advancement. Assuming two species have proof of a similar infection tainting them, it tends to be a sign that the regular progenitor of the two was contaminated before they developed into independent species. Once the infections become stuck in our genome anyway and passed on, they can effectively affect advancement. The infections generally become inactivated and reshuffled across the genome, and some of the time this blending can be worthwhile. Some human qualities utilize the advertiser districts of viral DNA to be actuated. Some have even been co-picked into the human resistant reaction framework, an instance of infections battling infections.